Welcome again to this NPTEL online certification course lecture on biological process design for wastewater treatment. So, today we will continue with the new stoichiometric reaction that we started studying in the previous class, the new section that we had started. So, in the last lecture we understood that there are two types of reactions which occur which help in the overall growth of microbes. So, those reactions were called as anabolic reactions and catabolic reactions. In the anabolic reaction, the carbon source is utilized along with the other elements to make microbes or microbial cell materials, and during this reaction we require lot of energy, and that energy is obtained from catabolic reactions in which the ATP is generated, which actually gets transferred into anabolic reactions where ATP is utilized and it is converted into diphosphate which is ADP, and ADP again transferred back into the catabolic reaction. So, this continues. Later on in the previous reaction we studied the stoichiometry of two of the reactions that happen during the anabolism. So, stoichiometry, we started studying the stoichiometry of anabolism, under which the overall anabolic reaction for microorganism was schematized as some carbon source, some elements they get converted into microorganism. For the microorganism we are using the general formula this, for the carbon source we are using a generic formula of CWHXOYNZ, and then we started understanding the metabolic reactions of heterotrophic microorganism under various conditions. So, first condition that we studied was aerobic condition. So, we studied the aerobic metabolism and we found out the reaction can be represented by this particular equation, where the carbon source along with ammonia and oxygen they react to form the microorganisms and H2O. Now, in the anoxic condition, when the oxygen is not available, so under that condition we use nitrate along with ammonia and certainly the carbon source, and they get converted again into microorganism, the nitrogen gas and the water. So, in place of in the anoxic condition nitrate gets converted into N2, whereas in the aerobic condition the oxygen is there. So, oxygen is used. Now, we will further study other types of metabolic reactions under the anabolism category, and later on in the today's lecture we will study the same thing under catabolism. So, we will study the difference. Now, for aerobic metabolism of autotrophic nitrifying microorganism, now we are going for nitrification. For aerobic metabolism of autotrophic nitrifying microorganism, the carbon source is carbon dioxide, where carbon has a oxidation state of +4. Therefore, this carbon will have to be reduced to be incorporated into the biomass, and for this the reducing agent is NH3. Now, nitrogen in NH3 has an oxidation state equal to -3 and it gets oxidized to nitrate which is having an oxidation state of +5. Therefore, the oxidation half reaction for this autotrophic nitrifiers growing on CO2 is this. So, this is there. So, ammonia reacts with water to form HNO3 with some electrons released. Similarly, in the reduction half reaction the CO2 is utilized along with the ammonia and these electrons to form the microorganism. Now, this overall combining both reactions we have CO2 which is getting utilized along with ammonia to form microorganism HNO3 and H2O. So, this is the overall aerobic metabolism of nitrifying microorganism. Now, in the fermentation reactions what happens? So, there is another type of reactions which are possible in the wastewater treatment, which are fermentation reactions. So, in fermentation reactions with an organic source which was the source that we have been using in the previous lecture also, there is no external electron acceptor, that is oxidant is not. In this case the electron acceptor is the H3O molecule or H plus ion, which is released in the during the oxidation of carbon source, and therefore, it can be said that in anaerobic fermentation reactions the substrate itself is both oxidized and reduced. So, the oxidation half reaction is same we have seen for aerobic anabolism. So, the this is the reaction. Now, this is the half reaction for aerobic anabolism. However, the reduced half reaction is the reduction of H plus ions to H2. So, this is the difference in the fermentation reaction. So, we have this reaction, and combining together we have the carbon source combining together with ammonia to form the microorganism H2O, and H2 gets released. Remember here H2 is getting released in the fermentation reactions. Now, there is another type of reactions which are possible in the biological treatment. These are called methanogenesis reaction, and this metabolism in the anaerobic digester where methane get released. There are two types of metabolism possible. One is called acetolactic and another is called hydrogenotropic methanogenesis. So, in the acetolactic methanogenesis the substrate is acetic acid, for example, which is produced with the catabolic reactions of many substrate. The anaerobic reaction of the acetolactic methanogenesis is only a particular case for any other reaction, but it can be represented by this particular reaction where acetic acid is reacting with ammonia to form the microorganism along with water. And during the hydrogenotropic methanogenesis, the carbon dioxide is used as a carbon source, and for the anabolic reaction carbon dioxide is reduced to biomass and the reducing agent is hydrogen. So, we have hydrogen which is used as the reducing agent. So, CO2 with 4 electron plus 4 H3O plus and ammonia, they produce the microorganism along with the water, and H2 along with the H2O release the electrons and H3O plus. If we combine together, overall reaction is this: hydrogen plus CO2 plus ammonia produce the microorganism during the hydrogenotropic methanogens. So, this is the overall reaction that happens during these particular types of reactions. So, we have studied few types of anabolic reactions, and overall their stoichiometric equations can be summarized here. First we studied the heterotrophs under aerobic condition, then heterotrophs under anoxic condition. In the aerobic condition oxygen was present, whereas in the anoxic condition oxygen was not present. So, we used HNO3. HNO3 was converted into N2 under anoxic condition, whereas oxygen was utilized as such during aerobic condition, and the reactions stoichiometry are given here respective by two reactions. Later on in today's class we studied regarding the nitrifiers, where we can see the carbon dioxide along with ammonia is used for the reaction. Similarly, during the fermentative process the carbon substrate along with the ammonia forms the microorganism along with water and hydrogen. In the hydrogenotropic, again hydrogen is used along with the carbon dioxide to produce the biomass. During the acetoclastic, the acid is used along with the ammonia to form the microorganism. So, these are the various anabolic reactions which occur during the wastewater where substrate is utilized for production of biomass. Now, there are catabolic reactions which actually provide the energy. So, now we will study the catabolic reactions in detail. So, stoichiometry of catabolism. So, we will start with this. So, considering the aerobic metabolism of heterotrophs. So, first we will start with the aerobic metabolism of heterotrophs on the organic substrate, which was same as earlier. The catabolic product is carbon dioxide and the oxidant is oxygen. So, for simple aerobic condition we have oxygen which acts as an oxidant, and this oxidant works on the organic substrate to produce CO2, electron and H2O during the oxidation half reaction, and during the reduction half reaction this reduced oxygen which was seen previously, it is utilized to produce water. And if we combine together both the reactions A and B, so we have overall catabolic reactions can be written as where some substrate is oxidized in presence of oxygen to CO2 and H2O and NH3 if nitrogen is as a element. So, already we have seen such type of reaction during our theoretical calculation of THOD. So, same is being repeated here. Here they are called as catabolism reaction because they provide the energy during the aerobic metabolism of heterotrophs. Now, going further, for autotrophs nitrifying microorganism, the catabolic reaction involves the oxidation of ammonia to nitrate using oxygen as an electron acceptor. So, this is used to produce the nitrate. So, here oxygen is there, ammonia is there. So, we have HNO3 we gets formed and plus H2O. During the anaerobic fermentative microorganism reactions, there is no electron acceptor, and therefore, it is similar to anabolic reaction which was shown earlier. So, in the catabolism reaction the substrate itself is both oxidized and reduced as it was the case earlier. The final oxidation product for catabolic reaction is very often carbon dioxide, and the reduced products are different depending upon the carbon source and the microorganism type. So, if we take like glucose is the substrate, the reduced product from the catabolic reactions will be ethanol or hydrogen, the hydrogen production being associated with production of organic acid. So, let us consider the case where glucose is converted to carbon dioxide, hydrogen and acetic acid. The oxidation half reaction for this case can be written like this, where we can see the glucose is being converted into the acetic acid in presence of water and CO2 is getting released. The reduction half reaction can be written like this, and combining together the glucose is getting converted into acid, hydrogen and CO2. So, this is the catabolism reaction, and in this reaction the energy gets released. During the methanogens, there are two types of methanogens possible, the acetoclastic and the hydrogenotropic. Now, in this acetoclastic methanogens, the catabolic reaction is the oxidation of acetic acid to carbon dioxide. So, here the conversion happens from acetic acid to carbon dioxide. The oxidant is acetic acid itself which reduces to methane. So, we can write the half reaction for this reaction, these two half reactions are written, and we can see the acid itself is getting reduced to methane, whereas in the oxidation reaction the acetic acid is getting reduced getting oxidized to CO2. So, both acetic acid itself works as both oxidant as well as both reduction and oxidation of acetic acid is happening, and we combine together both reactions to get 2 moles of acetic acid getting converted into 2 moles of methane and 2 moles of CO2, or simply we can write without 2 also. So, this is a straightforward reaction which can be written. Now, continuing for hydrogenotropic methanogens, the catabolic reaction is the oxidation of hydrogen to water. So, in this case with carbon dioxide being used as an oxidant. So, we can see here the carbon dioxide is being used as an oxidant in biological reaction, which is never possible in usual scenario. The oxidation half reactions are H2 combining with water to form H3O plus and 2 electrons, and in the reduction half reaction the CO2 combining together with 8 electrons and H3O plus to form methane and water. Now, we have to combine both reactions together, and for this we will have to multiply the first reaction by 4, so that 8 electrons and 8 electrons go off, and overall reaction can be written as CO2 plus 4 H2 produces methane plus water. So, this is the hydrogenatotropic methanogens reaction with under catabolism conditions. Now, the summary of catabolic reactions can be written here. In the aerobic condition, we have oxygen. In the anaerobic condition, we have nitrate. Oxygen is getting converted, all the carbon source is getting converted into CO2, H2O and ammonia under aerobic condition, and in the anaerobic condition, this HNO3 is getting converted into nitrate, H2O and NH3, and both these reactions are, one is occurring under aerobic condition, another is occurring under anoxic condition. Further on, there are other types of reactions which are possible under catabolic reaction category, and one is called nitrifiers, we already studied, and in the nitrifiers ammonia combines with oxygen to form HNO3 and water when the nitrifier microorganisms are present. When the fermentative microorganisms are present and we are representing glucose conversion to acetic acid, we can write like this equation, and we can see here that CO2 is getting produced. In another reaction, when acetoclastic conditions with respect to methanogens is there, so methane is getting produced along with CO2 from acetic acid itself. In the second case, when the hydrogenatotropic reaction is happening, so hydrogen is being used along with the CO2 to produce methane and water. So, till now we have studied anabolic reactions, catabolic reactions separately, and under various conditions when different types of microorganisms are present, and depending upon the availability of whether oxygen is present or not present, all the reactions have been listed. Now, the challenge is that can we combine together these reactions and both anabolic and catabolic reactions together and come up with a combined reaction. So, this is the challenge, and this is what we are going to study. And so, we want to study the overall growth stoichiometry. We will end with this question: can we write a stoichiometry for overall process of microbial growth? In other words, can we combine the anabolic and catabolic reaction to obtain the overall growth chemistry? Answer is, it is not possible to write an overall stoichiometry for microbial growth purely based on elemental valences oblique and or oxidation reduction reactions. Why this is so? In general, it is not known how much amount of energy is generated in the catabolic reactions and how much is actually being transferred to anabolic reactions. So, this is one of the basic reasons where it is not easily possible to combine together both. Second is that the energy is transferred from catabolisms to anabolisms via ATP and ADP, and it is very difficult to predict on paper the amount of ATP that can be generated per unit of energy generated by catabolic reaction. So, we can find out the energy generated by catabolic reactions, but it is not easy to find out how much ATP has been generated per unit of energy generated, and even for the same substrate and the product and for the same growth condition, the efficiency of oxidative phosphorylation varies greatly among different microorganisms. So, because of this condition, the overall growth chemistry is stoichiometry is not easy to understand, and we have to use certain other conditions to combine together. So, we will understand and we will start with little bit studying the overall growth chemistry in the next class onwards, and we will combine together the anabolic and catabolic reactions. There is a difficulty in solving them together. So, we will use a certain conditions under which they can be overall combined together, and then they can be used together for some inferences which can be derived out of the overall microbial growth stoichiometry. So, with this we will end today's lecture. Thank you very much.